Now I'm just testing the video and the sound on the stream. We should begin at around 11, and in the meantime I will be preparing but already streaming because I prefer to start this in advance. Now I'm just testing the video and the sound on the stream. We should begin at around 11, and in the meantime I will be preparing but already streaming because I prefer to start this in advance. Now I'm just testing the video and the sound on the stream. And apparently I do have 9 viewers, which is exciting, or even 12. Thank you for joining me. This is quite exciting. For those of you writing me wishes, I will be disabling notifications as to not distract myself, but I will of course read all of the messages in retrospect. And I would also note that communication should be possible among people in the chat which accompanies the stream. I will not see it, but if you feel like communicating with other people watching the stream, this may be an interesting exercise to try. Good morning. Good morning. I'm just handling Zoom in parallel with streaming things onto YouTube, which I think is quite important to mention for the members of the jury. We're public and live, and already have 20 viewers watching us. Yes, I apologize for that, but when I will transition between presentation and back, I might take a second to make sure nothing's broken, but in principle I'm just capturing one of my screens, and all of the sounds I can hear can be also heard by the public. Well, the presentation can be rewound, so if you're ready, I should be also. I think. Okay. Thank you. So. Mhm. Okay. Yes, I was about to propose that. Uh, yes, indeed. It is the English spelling rather than the French one. Okay. So I would also apologize in advance that because I have multiple screens, sometimes it would appear as if I'm not paying attention, but in fact that is because I'm either checking the stream on my right or the full screen slides on my left. I would expect everyone to be more comfortable with English than with French. Uh, I'm told that apparently people cannot hear you, so I'll just quickly tinker with the settings. Oops. This is unexpected and strange. So I think I need to switch to this, and now hopefully people can hear me.
I think they will hear you in any case, but maybe it's us.
Yeah, sorry, and you.
Mm-hmm.
But yes, it's because the setting has changed on Zoom a bit. It should be okay now, I hope.
Good. And it's exactly 11, so I will give the session to Gilles.
Okay.
And start the defense process.
Okay, so welcome everybody. So we are here for the defense of Vassili Chemchev.
Okay.
So the work is thèse de doctorat de l'Université Paris. I don't know what the PSL name is for actually, but the PhD was prepared at ESPCI, and the title of the work is towards biocatalyst selection using microfluidically distributed molecular programs. And maybe before leaving the stage to Vassili, all the jury members will present themselves, and starting with Karen.
Hello, I'm Dr. Karen Plizzi from Imperial College London. I'm in the Department of Chemical Engineering and in the Center for Synthetic Biology there.
Okay, then Philippe.
Hello, I am Philippe Lengue. I'm an assistant professor at ESPCI and working in the laboratory of biochemistry.
Okay, then after Yannick.
Hello, I am Yannick Landlez. I'm the mentor of this PhD work. I'm working at ESPCI in Paris.
Okay, and lastly, Gilles Truand. I'm working in Toulouse Biotechnology Institute in Toulouse. So first we will hear Vasily for about 45 minutes, and then we'll have a session with questions from the different members of the jury, and then we will go all together, the jury will go together to deliberate. Okay, so Vasily, it's your turn now.
Okay. Ladies and gentlemen, the esteemed members of the committee who've just been introduced, it is my pleasure today to present my doctoral work, which I have carried out under supervision of Yannick Rondelez. It was supported by the ERC, and in particular it was part of the PROF project handled by CNRS, and the work was carried out in the Gulliver Lab of ESPCI, which is now part of the Paris Sciences and Lettres University at the École Doctorale AMTCI. And it is indeed titled Towards Biocatalyst Selection Using Microfluidically Distributed Molecular Programs, which might sound like a lot of unfamiliar words to the general audience. And therefore, I would like to start with some general introduction to the biochemistry, which I have been studying for quite some time, making this title a little bit more obvious to me, which would intertwine with some of my personal motivations for this project. I would then cover some of the prior art, which is relevant to my work, and present small molecule sensing in pen-DNA molecular programs, which is the core element of my project, as well as some of the potential applications of sensing for directed evolution, and then speculate about potential future work. So, in a very broad overview, during my undergraduate in biochemistry, I've spent quite some time studying that DNA, RNA, and protein, and as the central dogma of molecular biology suggests, DNA replicates the genetic information within itself, which is then transferred to RNA, and finally into proteins, which somehow represents the all of the activities which happen within our bodies. If you're studying biochemistry, you study in quite a lot of detail that desoxyribonucleic acid, which is the full term for DNA, is a long and stable polymer which contains genetic information encoded by nitrogenous bases such as adenine, thymine, cytosine, and guanine. And in living matter, these are usually present in a pair of two strands, which facilitates replication of genetic material. The properties of these base pairs is such that cytosine always prefers pairing with guanine and thymine with adenine. And therefore, if you only have one strand out of the two, you can easily fill in the complementary one thanks to the particular hydrogen bonding. This specificity in interaction allows us to also program interactions of short DNA strands, which we can synthesize artificially, and therefore govern behavior of artificial systems which are DNA based, and I will talk about this in a bit. The second thing biochemists spend quite some time on is studying proteins, and in particular enzymes, which are usually made of several chains or one chain of amino acids, where one of 20 naturally occurring amino acids is defined by 3 nitrogenous bases. We can immediately see that 64 combinations could be possible, so there is some redundancy, as well as some of the triplets coding for a stop signal in translation. And the exciting thing about enzymes is that they can significantly increase reaction rate, and therefore this is described as enzymes exhibiting catalysis. And in natural systems, a given chemical reaction would usually be occurring so slowly that you could consider it negligible. However, if an enzyme is present, the reaction is taking place, and a collection of chemical reactions which we have in our body could be described as metabolism and philosophically life. frequently, these are named after the activity they exhibit, followed by an A suffix. So, for example, an enzyme acting on DNA would be a DNase. And having looked at enzymes and DNA for 4 years out of passion and pure curiosity, I started wondering how I could make a living with it and be valuable to society. And as some pop culture suggests, you frequently do things and then only start thinking of how to proceed next. So in my case, I got my bachelor's in biochemistry, I got my master's, and I was thinking of how to make a living with it. I've tried some teaching, which results in more people knowing about DNA and proteins and then being able to spread this knowledge. But luckily for us, there is a more conventional way of making a living in taking a certain material, doing something to it to add value, and then maybe passing it on at a higher price. And this is possible in biochemistry. I've recently found a notebook from my granddad, and it seems he was sometimes doing exactly that. And we can see that he's taking some chemical molecule which has a certain price to it, and then converts it into a different molecule which has a different price per kilo to it. And most importantly, we generate a profit, which allows us to sustain this as a business and provide people with a cool molecule they use. And at the heart of this is, of course, an enzyme which enables this conversion. Since the times of my granddad, this idea is older than I am. Things have changed a little, and some more simple molecules have already been produced. So we're thinking if we could do something similar, yet modern. And a common target for studies these days is tryptophan synthase, and in particular tryptophan synthase subunit B, which in nature allows the synthesis of L-tryptophan by combining L-serine and indole. However, it has been noted that you can feed it alternative substrates, such as halogenated indole moieties, and then, although there's some reluctance, the tryptophan synthase subunit B would produce a halogenated amino acid, which can have many applications in both research and medicinal applications. So, if we quickly check if this is indeed relevant, we can go on our favorite chemical supplier website and look up at the prices of the substrates and the potential products, and quickly check that L-serine goes at around 1,000 euros per kilo, indole 800 euros per kilo, and then L-tryptophan at just over 1,000 euros per kilo. So the business plan here would not look too convincing. And this is in part because L-tryptophan is not sourced through artificially combining L-serine and indole, but rather extracted from some other bioproducts. However, if we look at fluorinated indole, we notice that suddenly the stakes are much higher and the commitment to an enterprise would be significant. Yet, in my opinion, it could pay off, as fluorinated L-tryptophan goes at around 3 million euros a kilo. Here, I think this could be quite an interesting enterprise. But an important message here is that nowadays we have a demand for non-natural molecules, which in turn needs non-natural enzymes. As I've mentioned, someone has already ventured in this direction, and in particular the group of Frances Arnold, who's recently received a Nobel Prize, worked extensively on trying to improve tryptophan synthase subunit B to make it work on its own and to allow it to accept artificial substrates. And their approach, which was recognized by the Nobel Prize, is termed directed evolution. So I think it is quite important to look at that. Directed evolution is somewhat similar to natural evolution in that some object is taken. However, here this is done on a molecular level, so it is a gene coding for a particular enzyme. The genetic variants are then diversified through some form of mutagenesis, and the corresponding enzymes are expressed in such a way that a correlation between a particular variant of the enzyme and the DNA is maintained. After that, some form of screening or selection, as defined by the experimenter, can be applied, and better suited variants can be selected and amplified. The cycle can then continue with increasingly better variants being sourced. This is depicted on the right, where starting at a given point, one would gradually push variants to become more and more fit. Where fitness is defined by experimenter, it could be stability, it could be acceptance of a certain substrate. More trivially, it could be the rate of a chemical reaction, gradually reaching some local or absolute maximum of fitness you could expect from a particular enzyme. And quite a lot of attention is paid to the screening and selection stage to find these good variants. So in case of Frances Alner, they've shown that you can find a variant which is 20 times more catalytically efficient than the wild type by screening 528 clones. Now here I'm illustrating the concept with some GFP work I've done before, which is not exactly related to directed evolution. But to general audience, I would note that some undergraduate student would have to go and by hand pick individual bacterial colonies, seed them on a 96 well plate containing 100 to 200 microliters of media for bacterial growth here, and then using some machinery screen these wells for better variants. Now here, because GFP is fluorescent, we can more or less see by eye that some variants are more successful than others. If you're screening for an enzyme, you have to express this enzyme within bacteria, somehow extract it from bacteria, supply them with the reactants, and monitor the chemical reaction in real time to observe the reaction rate. And if you want to screen 528 clones, you would need about 6 of these 96 well plates. Can we increase the throughput and be more economical on both time and reagents? The answer is yes. Currently, microfluidics, and in particular droplet microfluidics, which allow generations of emulsions, are coming into fashion. Here you can generate over millions of samples per hour, which are only picoliter in volume. However, you still need to somehow sort the better variants, as illustrated here by this device, and this is done by one with a backlog of sample previously prepared, and it is a twofold bottleneck. First, you have to consider the rate at which you can sort these droplets one by one, and secondly, you need a reactant, so either a substrate or a product, which is detectable by this laser here, so it has to absorb all fluoresce. So it would be good if these droplets somehow magically selected themselves and then amplified the good genes and ignored the bad ones. This idea first occurred to Philip Holliger in a process which he called compartmentalized self-replication, which, as the name suggests, involves a selection of an enzyme which can replicate itself. Here, a library of thermostable polymerase variants is taken and incorporated into bacteria, which are then compartmentalized in emulsion together with desoxyribonucleoside triphosphates and primers that enable DNA amplification via PCR later. The DNA polymerase is expressed within the cells, which are then lysed during PCR cycling. And if the DNA polymerase is good, it can amplify its own gene efficiently and propagate it within the library. Whilst if a particular gene for a DNA polymerase is not so good, it will replicate its own gene less well and therefore this variant would not be replicated in the library. Within our group, this was taken a step further to try and generalize this approach to other enzymes, and in particular, I'm following in the footsteps of Adel Dramain, who was the first one to attempt pen compartmentalized self-replication, followed by Rémi Siskind, who's moved this procedure in vitro. The consideration for in vitro work is that in case you want to evolve an enzyme which activity could be toxic to the cells, or if you're working with unnatural compounds which could somehow be incompatible, you avoid this by removing the living organisms you have to work with. And the schematic here, although it might seem rather complex, is in essence very similar to the two we've seen before. There is a library of genetic variants somehow prepared, and the DNA as well as the corresponding protein is linked on a solid bead support, which is then subjected to some form of selection with this magical pen toolbox, which I'll talk about in a second. I would also note that they worked primarily on cases, and it will soon be obvious as to why. So if we look at this part closely, which represents the self-selection step, we can probably summarize this process in this diagram here. You have DNA, which is corresponding to a particular enzyme variant. This enzyme demonstrates some activity. In case of both polymerases I've shown before, and nicases, which are a group of enzymes which incorporate a single-strand break in double-stranded DNA in a defined recognition domain. These impact DNA, and since the pen DNA toolbox I'll present on the next slide is also based on DNA, there is an immediate interaction which leads to production of primers, which can then be used for PCR, which facilitates DNA replication. And therefore we'll have a closed loop system where good DNA would produce a good enzyme demonstrating good activity and propagate all the way to replication of the DNA. However, coming back to the idea of tryptophan synthase, our activity would be production of small molecules, which do not immediately affect DNA, and therefore we need an interface which would connect small molecules to our pen toolbox. Without further ado, I would introduce the toolbox now. The pen DNA toolbox consists of a series of modules which are called templates and are short strands of DNA driven by such enzymes as DNA polymerase and nicases. For the majority of it, it would be NBBSM1 as well as NTBST-NBI. So an example of a pen DNA toolbox module is a source template I'm showing here, which is a hairpin structure containing an NTBST-NBI recognition domain and a distal cleavage domain, as well as some output sequence, which we will call trigger sequence, or in my case also B11, because it contains 11 base pairs. And due to the nicking site, the NTBST-NBI can come, introduce a nick, and produce a short fragment which can then dissociate from DNA and proceed to other modules of the toolbox, whilst the polymerase would recognize partially double-stranded DNA, elongate it, therefore allowing the nicking to occur again, resulting in a linear production of this signal. The next module can be the autocatalytic template. It can accept the trigger sequence due to partial complementarity. The sequence would then be extended to contain the sequence identical to the trigger, and the combination of two of them would form a nicking site in the middle, which can be recognized by NBBSM1. Nicking occurs, resulting in two short identical fragments, both of which can dissociate and participate again in this process, therefore gradually leading to exponential amplification. A third module which would be relevant to this talk is the pseudo template, which similarly to the autocatalytic template can accept the trigger sequence. However, it is fully complementary and therefore has higher affinity for the trigger. And when trigger is extended, it is extended with a short junk sequence such as a poly A or a poly T. Therefore, once it dissociates, it can no longer participate in the autocatalytic cycle. These modules can be combined together, and if only a small amount of trigger sequence is supplied, due to high affinity, all of it will be bound by the pseudo template, become inactivated, and will go to waste. Therefore, the pseudo template module represents a sink of sorts. However, if a certain threshold is exceeded, the overflow of trigger would go to the autocatalytic module and activate exponential amplification. Therefore, this system is bistable, and for convenience, we prefer monitoring the state of the system using the reporter template, which is another hairpin structure containing a fluorophore and a quencher next to it. Upon trigger sequence invasion, which is then extended, this hairpin opens up, and once the quencher is distant from the fluorophore can fluoresce, and we can monitor the signal. Alternatively, we can of course take our trigger sequence and, a little bit like with the autocatalytic template, use the primer producing template to extend this sequence into a primer sequence, which would then be cleaved off and released. And here we can linearly produce primers, which would close our loop for DNA replication. I would recap this by reminding you that the assembly of the autocatalytic template, pseudo template, and reporter template allow us to observe the bistable switching behavior, so I would refer to this assembly simply as the switch. And also, in principle, the combination of these templates allow us to take enzymatic activity such as anti-BST and BI, which is why prior work focused on this, propagate the signal and amplify it if it exceeds a certain threshold, produce primers, which can then be used in DNA amplification. However, it is not trivial how small molecules could contribute to this process. So if we look at Nature for inspiration, transcription factors are commonly used to regulate DNA expression. In this work, I've used lactose inhibitor and tryptophan repressor. Lactose inhibitor binds a particular sequence of DNA called the operator sequence, and prevents RNA polymerase from being able to bind and proceed through DNA for transcription. However, if an inducer molecule such as IPTG or allolactose is added, conformational changes occur in LAC-I upon binding this inducer molecule. It dissociates from DNA, and then transcription can occur. Tryptophan repressor works similarly, yet there is an important difference that it is unable to bind DNA unless the repressor molecule, such as L-tryptophan, is present. Therefore, if we summarize the behavior in a simple glyph diagram here, IPTG activates transcription through the LAC-I inhibitor, whilst L-tryptophan represses transcription through tryptophan repressor. If we incorporate this operator site within the source template, we can envisage a design such as this one. As before, we have the nicking site, which leads to production of the trigger sequence, which can then participate in the switch. However, we also have the transcription factor operator site, which can be bound by the transcription factor, and as anti-BST and BI has a cleavage site which is 4 base pairs away from the recognition domain, we would expect a steric overclash and therefore interference of the transcription factor bound with anti-BST and BI ability to nick and produce the output. However, upon addition of the inducer, as in natural systems, we would expect the transcription factor to dissociate and the cycles of sequential nicking and polymerization to take place to linearly produce the trigger sequence of interest. These can then go towards the switch to activate the system. In a static form, we would recap this in this diagram where upon inducer addition, the transcription factor dissociates and output is produced. But first we need to check that indeed transcription factor binding such as LAC-I interferes with the production of the output. And I think it is a good opportunity for me in parallel to this to demonstrate the type of data we usually obtain and how we treat it in these 4 steps. So diagram A is the raw data where we observe over time a series of samples colored from red to purple in decreasing concentration of LAC-I dimer against the fluorescence exhibited by the Sci5 fluorescent dye. And we can see here that at the beginning the fluorescence is stably low. And then upon switch activation, there is exponential amplification leading to probe opening. And once the reporter template is fully open and there is no more fluorophore to dequench, there is another plateau reached. As these plateaus are slightly uneven due to some discrepancy within the machine, we prefer to normalize these curves between 1 and 0 by minimal and maximal fluorescence, which we can see in diagram B. From here, we can then extract the gradient of individual curves, which is plotted in C, still against time. This allows us to both observe whether the gradient and therefore the rate of exponential amplification changes between the samples, and also to determine the point of maximal gradient, which we define as the CQ value or the switching time. And we can then plot the switching time against our LAC-I dimer concentration in this plot here, obtaining some sort of trend. We find it more convenient to plot 1 over CQ values instead, which I will show you on the next slide, because then intuitively the fastest systems correspond to higher numerical values. And also in case a particular sample has not switched at all, you can assign it a value of 0 rather than infinite time of switching. So without further ado, if we plot 1 over CQ, we can see that higher concentration of LAC-I dimer leads to slower switching time, which is in line with our expectation that it would bind the protein sensing template and repress production of the output. It is also worth noting that here we used 5 nanomolar of the protein sensing template, and at around 4 nanomolar of LAC-I, we see the maximal repression, therefore suggesting the expected stoichiometry of 1 to 1. Building on this, we of course attempted to detect IPTG, which is a frequently used inducer for the system. And we can see that if there is a saturating amount of LAC-I which represses our switch, and we start adding an increasing concentration of IPTG, we see this typical sigmoidal induction curve leading to gradual derepression of the system and faster switching. As a control, we used lactose, which is structurally similar to IPTG, and yet it shows no effect on this sensor. I would use throughout the talk quite extensively these little glyphs summarizing the logic of the system. And for the LAC-I based protein sensing template, the inducer activates the switch. Similarly to IPTG, we can use the allolactose I've mentioned before as a natural inducer of the system, and we can see the same sigmoidal trend. What's interesting here is that lactose and allolactose are very similarly chemically. It is only the difference of 1 to 4 and 1 to 6 glycosidic bond. And yet there is a response to allolactose and there is no response to lactose. Now, if we try using the same architecture with tryptophan repressor, we can do this. The only difference would be that here the output would be produced unless the repressor is added, in which case the system is repressed. This is pointed out with this step of the logic flow. So the repressor represses the protein sensing template, which by default would be producing the output to activate the switch. Summarizing, this repressor represses the switch through PST. And indeed, we observe this in an experimental result with increasing amount of L-tryptophan gradually repressing our system, whilst L-threonine, a somewhat similar amino acid, having no effect on the system. Coming back to our idea of selecting tryptophan synthase subunit B, we would probably want to encourage a variant which is fast at producing L-tryptophan, and therefore higher concentrations of L-tryptophan should correspond to faster switching and earlier primer production. For this, we need to invert the signal relayed by the transcription factor, and we propose to do this by a system which might seem a little bit complex at first, but if we compare it to the one I've just shown you, there is still this component of a sensing template which can be repressed and which produces a given output. However, in this case, the output is labeled as anti-B11A due to the fact that it is fully complementary to the natural B11A output, would bind it, and they would mutually annihilate themselves within the system. Therefore, anti-B11A acts as a form of sink. In parallel to this, we have our normal switch assembly, which would attempt to start due to a small amount of source template, always linearly producing a trigger trying to push the switch to activate. This would be quenched only if anti-trigger is produced. This can be summarized in this logic diagram. On the left, we have the bistable switch as before, which is constitutively activated by the source template. And on the right, we have the repressor, which would be expected to repress the protein sensing, now killer template, as we termed it, because it kills the switch. And it kills the switch through production of anti-trigger, which would consume normal trigger. Therefore, now compared to the system I've just shown you, where the repressor represses the switch through the protein sensing killer template, the repressor would now activate the switch. If we attempt this experimentally, we do indeed observe that increasing concentrations of L-tryptophan do lead to faster activation as we would like to. For the interest of completeness and to see if we can propagate this strategy to other transcription factors, we've also inverted the LAC-I system. As before, it is similar to the protein sensing template for LAC-I, with the difference being that anti-trigger is produced and it quenches the switch which would otherwise be started with a source template. The logic diagram here on the left is identical to the one I've just shown you, and on the right the only difference is that now it is inducer which activates the protein sensing killer template to then quench the system. Therefore, instead of the inducer activating the switch, the inducer represses the switch through the protein sensing killer template, and in line with our expectation, now increasing concentrations of IPTG repress the system. So we have this toolbox of sensing templates which allow us to invert the logic in any way we would like to. How can this be relevant to directed evolution? Well, in principle, you can expect an enzyme which would produce both the wild type product you do not want and also display some promiscuity to produce the artificial, non-natural new products you would like. And you can imagine that with this combination of sensing templates, you could penalize this enzyme if it produces the undesired biomolecule, and contrary to that, activate the switch if the desired product is made. If we try using a combination of two inputs, we can arrive to plots such as this one. Here I'm using two protein sensing templates, one responsible for IPTG sensing and one responsible for L-tryptophan sensing. And we can see a little bit like before that with increasing amount of IPTG, the system is faster to start. whilst with decreasing amount of L-tryptophan, the system is also faster to start. And we can note that in a certain combination of concentrations, the response in both directions is symmetrical, and therefore we can have an even weight to two inputs. We can, of course, make all possible combinations of the sensing templates, resulting in these four rather rich plots, but the main take-home message here is that you can see a degree of rotational symmetry between them. And we can also note that the input is reasonably even between the two small molecules, therefore they can be balanced. I've shown you before the two protein sensing templates, but here we can use two protein sensing killer templates, and now the response is inverted. With increasing amount of L-tryptophan, the switch speeds up, whilst with increasing amount of IPTG, the switch slows down. You can, of course, mix and match. For example, here, with a protein sensing template for LACI and a protein sensing killer template for tryptophan repressor, you can see that L-tryptophan activates the system, and IPTG also activates the system. And finally, this can be inverted, where both IPTG and L-tryptophan repress the system. Now, although here I'm showing an even response, which is reasonably gradual, this can also be altered. So if we take this particular plot forward, which is now shown here in A, and increase the concentration of the pseudo-template, which represents a threshold module, from 1 nanomolar to 4 nanomolar, suddenly quite a lot of samples are cut off and unable to start because the threshold is too high. An insufficient amount of input is produced. We can then try mitigating it in an alternative fashion by increasing accordingly the protein sensing template concentration, which would push the switch to start. In this case, we regain a function. However, now the balance of the protein sensing template to protein sensing killer template acting in another dimension is uneven. And whilst we can see a good difference between the samples in the L-tryptophan direction, in the IPTG dimension, the difference is not as noticeable. Hence, we can bias our system towards one of the two inputs. And finally, I would like to note that there obviously exists a set of conditions where the system starts to struggle to discriminate altogether. So our results so far involve the PenDNA toolbox as before. However, we can take, as previously done by other works, however, we can take a small molecule, plug it through some interface to have an effect on DNA, which then affects the pen processing and observe some fluorescence. A step further would be to add an enzyme which would have in situ activity and produce a small molecule which would then be detected. And with the help of my intern Camille Lescan, we have done exactly that. We have added to the system I've previously shown, namely the tryptophan sensing protein sensing killer template. We have added tryptophan synthase subunit B with a cofactor PLP, as well as L-serine and indole, which are the reactants, in hope that L-tryptophan would be produced in situ and activate the switch. In particular, we have taken tryptophan synthase subunit B from E. coli, labeled here as ECB, as well as two variants from an extreme thermophile Pyrococcus furiosus. One is wild type, and another one is a variant reported by Francis Aldred, which I've mentioned before, which is expected to perform better. And in line with our expectation, at 37 degrees, the E. coli variant outperforms the improved Pyrococcus furiosus variant, which in turn outperforms the wild type. We can also see that increasing concentration of the enzyme, which we would expect to correspond with increasing activity, in turn relates to faster start times. We think this simulates a situation where a better and a more active variant would start the switch faster and be able to replicate itself. However, for replication, we need to produce primers and carry out PCR. So we've also just done just that. So here is a rather data-rich slide, which I will talk you through step by step. On the left, we see the isothermal amplification of the PEN-DNA toolbox and the start times plotted as before. In particular, we have a range of E. coli tryptophan synthase subunit B from reasonably high to reasonably low. And we can see that as you decrease the amounts of the enzyme, the start time slows down. We claim this is because L-tryptophan is not produced as quickly. We also had a sample which had extra empty volume so that we could add primers for PCR control later, and a positive control containing 1 millimolar of L-tryptophan, which starts faster than the negative control, however not as fast as some tryptophan synthase reach variants, because here more than 1 millimolar of L-tryptophan could be produced. Moving to the right, we then kept exactly the same samples in the same machine and switched them into PCR cycling mode. And we now observe that, in line with expectation from the left, high concentrations of ECB correspond to faster PCR amplification. This decreases with concentration, with the negative control being the slowest to start, yet it does amplify a little as it also started at the primer production step, with positive controls being faster than the negative control, but in line with the slowest enzymatic samples. And first, to show you a snippet of the gel, we observe the expected 900 base pair fragment. For most variants, one could claim that it is not here for the negative control. And for some concentrations of the tryptophan synthase subunit B, the band, I would claim, is very similar to our positive control. Now, because I could not fit everything onto one slide, I would now move on to showing you the full gel, because I do expect some skepticism on excised bands. And here we can see that the bands are not dislodged too much. Something we also observe is high molecular weight DNA smears, which we attribute to excess of primers leading to concatenated DNA. Now, to recap the slides, I believe I demonstrate that we can take an enzyme which exhibits some activity related to small molecule production, through an interface influence PEN-DNA toolbox to produce primers as a function of activity, and then carry out PCR to replicate DNA. All of this in one pot, in the same tube, in the same machine, with little participation from the experimenter. Therefore, more broadly, I would like to conclude that hopefully in this presentation I've demonstrated that transcription factors can be detected with PEN-DNA. Small molecules can be also sensed by PEN-DNA. This sensing function can be modulated and inverted. Then L-tryptophan synthesis can be detected in vitro. And as I've just shown, amplification of the gene is also possible in response to enzymatic activity. Furthermore, I would like to add that the findings I have just presented are currently under revision in the nucleic acids research, where we have submitted a paper together with Camille, as well as Guillaume Gines and Yannick Rondelez. In terms of considerations for future work, of course, integration into the full in vitro PEN-CSR cycle is expected. I have no doubt it will be functional, as both Adèle and Rémi have demonstrated all of the other elements to work. And I would just like to show you some preliminary results that an enzyme which is immobilized on the beads can also demonstrate activity, and this activity can be detected. So if we come back to the workflow proposed by Rémi, you expect this enzyme to be stuck on the bead, and one could think that this would somehow interfere. However, when I immobilized E. coli tryptophan synthase subunit B with a snap tag on beads and added various amounts of these beads onto my PEN sensor, I was still able to detect enzymatic activity and amplify DNA, and we can see that higher concentration of beads, which corresponds to higher quantity of the enzyme, in turn correlates to faster DNA amplification, whilst controls based on green fluorescent protein, which is snap tagged, were not amplified because they exhibited no activity, did not produce L-tryptophan, did not produce primers, and therefore PC amplification failed. I would also briefly note that PEN modules could of course be optimized to improve overall performance, and within the thesis I demonstrate quite a lot of different attempts at how this could be done. There is not enough time to go through all of this, but I would just produce a really short example that primer producing templates can be made shorter so that they are only activated once all of the other components have already activated. And here, if we compare the primer producing templates I've used in the work presented to those which are one base pair shorter, you can immediately see that lower concentration of L-tryptophan can now be detected, and this detection happens much faster, which in turn translates into more efficient DNA amplification. And once again, lower concentration of L-tryptophan is still detected and DNA is amplified in response. With this, I would like to thank you first and foremost, Yannick, who's the mastermind behind this PEN CSR workflow and project, as well as Guillaume Gines, whose expertise on molecular program allowed me to make sense of what I was doing. I would also like to hugely thank Rémi Siskind because he's developed this intricate in vitro PEN CSR pipeline and helped me find my way around the lab and taught me where things are, helped me order stuff and set up my experiment. As well as I would like to thank Gabriel Ragala and Camille Lescan, who were interns with me and were there for some of the pivotal moments in the project. And Rocio Espada, who sat with me for over 3 years and gave me day-to-day general counsel on how I should proceed. Of course, this leads on to thank of the broad molecular programs and systems team, with which I had so much fun over the years. And there are numerous pictures in which we can find Rémi, Camille, Rocio, Yannick, Guillaume, and Gabriel, if you just give me a second, is over here. But of course, the SPM team is no more, as we're now living in one big happy Gulliver family, whom I would also like to thank for all of the conversations I've had both about my project and just words of encouragement and shared coffee. In principle, all of the people I've reached out to and invited for this presentation all helped me and supported me through these past 3 or 4 years. So if you're watching this, thank you. I would also like to remember people who are no longer in the lab, as well as other important members, for example, Igor, who did not make it through the project. And with this, I conclude. Thank you.
Thank you very much, Vasily. Just one quick word to all. Of course, it's more fun to have everybody on site, and I'm sorry that the conditions, the sanitary conditions, cannot make allow to have the jury members and the public all together in the same room. It's terrible, I know, but we'll have to proceed that way for I think quite a long period of time. So. So we'll start duly with Karen asking you questions, but I'm also telling the public and even the members that if you want to intervene, well, the jury members, if they want to intervene during each of the questions from each jury member, of course it's allowed, and even if we have prolongations on one question, it's better to ask them immediately. So Karen, I will leave you the floor, sorry, and please go.
Okay, thank you. Thank you, Cecilia. Very nice presentation. Gives a very good overview of your work and very clearly presented. So I have a general question to start, which is that I could think of ways that you could, if you wanted to do directed evolution in a system that produces small molecules, directly interface the transcription factor with an output like a polymerase or a primer or something like that. So the pen system in between, does that give you additional capabilities that you think are worth it in order to have these extra mechanisms in your system?
So I think no matter how you would try to interlink it, one could obviously think of implementations in vivo, for example. You would have some constraints, and I believe Pen gives you the additional leverage of modularity and control of the selection function. I've shown you a particular example of how things are done, but you can of course think of exponential amplification of the primer. We can easily move the threshold of activation. We can plug and play transcription factors, something I have not mentioned, but obviously to produce the 5-fluorotryptophan instead, you would need to evolve the appropriate transcription factor, which is thankfully available. And I think the general message here for Pen is that you can have exquisite level of control as well as the plug-and-play modularity at the same time.
Okay, good. So could you talk a bit more about moving the threshold and how you would implement that? You can talk either generally or in a specific example if it's easier.
So an example I've covered in brief is the one I show in a two-dimensional system. Obviously, all of the components of a Pen toolbox, and usually it's a 20-something reactant mix, is the pseudo template, which imposes a threshold below which no starters occur, no matter how long you produce the input for, because all of it is consumed by the sink. So you can. It is always a little bit complex because you need to juggle different modules, but the way you normally construct it is that you have the protein sensing template in sufficient concentration to overcome this PT resistance. However, as you start repressing it, suddenly you're below the threshold. And then when you start de-repressing it back through the inducer, you hope that at some point you jump over. Of course, something I also show in my thesis is that you can mediate the sensitivity by varying the concentration of the transcription factor. As with increasing concentration of transcription factor, you'll have more repression and the kinetics will shift, increasing your sensitivity, but at the cost of having to produce more protein. Similarly, you can make the system more sensitive by decreasing the threshold and decreasing the amount of the protein sensing template at the same time. However, the system can then become a little bit too sensitive to repeatedly carry out experiments and produce consistent results. So I would mention that, of course, the way these 4 experiments were carried out were from D to A, and in practice there were maybe another 8 in between to try and find the stable window of observation where also both inputs would be even.
Thank you. Actually, that leads me on nicely to one question that I had, because I'm not as familiar with the Pen system. So the pseudo template, I guess, is to guard against unwittingly activating the switch.
Yes.
What causes that activation?
So there are a couple of things to be concerned about. So if we go here, primarily 2 things occur due to, let's say, unexpected activity of the DNA polymerase. One, and this is the primary concern, which is observed in other isothermal amplification systems such as XPAR, is that it can sometimes self-prime, and you can see that in presence of just the autocatalytic module and in absence of the pseudo template, as soon as you've accidentally self-primed once and produces a couple of triggers, you are inevitably having the exponential explosion, which poses a problem with the detection limits and makes the system generally less convenient to work with. And then a second consideration, which is of less relevant concerning to PT, but we find that it usually helps as well, is that the polymerase can sometimes erroneously produce some aberrant fragment of DNA, And then you have this gradual amplification of a messy ball of DNA which self-primes and propagates. And somehow the PT sometimes helps with this as well, presumably by quenching these non-specific fragments and also destining them for degradation. But primarily it's the first aspect of it.
Okay. So in that case, then, when you showed sort of your typical data, is the switching time consistent for a given template? So, for example, you showed in the beginning how you analyze the data, which was really helpful for me.
Yes.
So sort of the time where the switch starts to function for any given condition, is that the same from experiment to experiment?
If you fix all of the components, absolutely. So for example, when I had to produce the two-dimensional plots, I have produced large stocks of all of the component I envisaged needing. And then through extreme care, replicating the procedure from time to time, pipetting in exactly the same order with the same air humidity and temperature. I'm exaggerating, but it was quite close to that. You can replicate results. However, usually because enzymatic stocks change or your dilution of the DNA stock can change from month to month, we prefer running internal controls. So you always have a negative control, which is expected to stay stable for a certain window of observation, and a positive control, which you expect to denote the fastest the system can switch. And then you align all of your samples in between. So for example, here we can see that this is the fastest our switch can perform. And this defines our window of observation on the left. And in a good experiment, you would expect the samples to plateau off here. Unfortunately, I only have a 0, which I have not plotted here, but it does indeed plateau off.
Okay, and if you have a system without a sufficient amount of pseudo-template, do you see sort of a stochastic point at which time it starts to switch?
So this is similar to the moments where the pseudo-template becomes inefficient due to inappropriate temperature. And if I could quickly drop in a diagram from my thesis, I could probably illustrate that, for example, here. So as the pseudo-template becomes less efficient or is overwhelmed, you struggle resolving your positive and negative controls, because now the system can self-start too quickly for the intended start due to the input you've envisaged to occur. And you can think of it, so as I've shown you in the previous diagram, you have the fastest the system can switch. And if your self-start starts approaching this point, your window of observation constricts.
Okay. That makes, so it always manifests then as self-starting too fast, not, randomly self-starting at some point.
Usually, if your mix is consistent, the self-start occurs at the same point. And for example, for the, again, two-dimensional plots I've been showing, I assembled the system to be good enough, and I aimed for it to be fast so that I could carry out experiments consecutively within a 12-hour framework. So the system always started at around 800 minutes, which I think corresponds to around 14 hours. But I knew that all of my intended activation would be before that, and therefore the window of observation would be sufficient.
All right, that makes sense. Thank you.
For if you value sensitivity over fast experiment, which some of my colleagues do, which they run an incubation for 2 days, you would add less DNA polymerase, which is the problem with the self-start, the main cause of it, and just wait for longer. So I use BST polymerase, which large fragment, which is known to be more prone to self-start than Vent. However, we found that it is much more efficient at 37 degrees than Vent-XO minus polymerase. And then similarly, although I did a range of BST polymerase and I could afford a lower concentration, this would have slowed down the system. And I've picked the one which was just stable enough. So if we go higher, in fact, self-start occurs roughly at the same time as the intended start. I'm sorry, I'm getting a little warm here.
Okay, so that also leads nicely into one of the other questions I had, which is, in order to get all of this to work, which you didn't address in your presentation, you had to redesign the initial switch so that it functioned better at a lower temperature, correct?
Yes.
So in the process of doing that, I mean, to get an idea for someone who doesn't know the system, was it sort of obvious how to design such a switch? In terms of which elements go where, how long various sequences are? Can you use tools to predict, melting temperatures in that process, or is it more of a trial and error kind of?
It is a combination of both. Thanks to help of Guillaume Gines, as I've mentioned, I could rely on his expertise of designing 5 or 10 of these switches, although mostly for 50 and 45 degrees. So we did guesstimate in that we've removed 1 base pair. Usually the sequences are 12 base pairs, ours is 11. We then picked a system with a reasonably low melting temperature. I think one of the lowest ones offered. Of course, we're constrained by the nicking side we have to incorporate in between. And then we've just picked 2 at random. So in my manuscript, I talk about 2 switches. And in fact, originally we ordered 2, expecting at least one of them to function. Then you arbitrarily take the autocatalytic template at 50 nanomolar just because of the convention we use. You need the reported template at between 50 and 10 for it to be detectable in emulsion, preferably at 50. This imposes some constraints. For the enzymatic mix, I was lucky to originally inherit the one from before. And then you can vary concentrations of the enzyme 1 by 1 to see if you are indeed in the optimal state for yourself. So for example, I found that for me, I need a much lower concentration of NT-BSD and BI, something like 10 to 20-fold less of the enzyme, presumably because at the lower temperature, it becomes much stickier on DNA and struggles to dissociate. And then you can also vary the PT to arrive to a point of bistability. So I think in my... can you see the manuscript I'm showing? Yes, yeah. Right? So for example, here most trivially, you just keep on adding PT until you knock off the self-start, and then you usually try staying as close to this bistable point as possible because this allows some sensitivity. Although in my case I could have gone with 6 nanomolar, but I've chosen 7 to just be on the side of caution.
Okay. And in all, how long does such a development cycle take?
Depends on luck and on how picky you are. So I think I was getting my first results more or less immediately. And one of the problems I had with presenting the paper and writing the manuscript is that you find that experiment from experiment, you have a gradual evolution of your mix. So it is very difficult to present a succession of experiments with consistent conditions. And in fact, I'm quite proud that my method section is so short because I managed to cherry pick experiments with the same mix. And for chapter 1, you can see that the figures are all quite nice and pretty because throughout the 3 years of my PhD, I arrived at a mix I was comfortable with, and then I fixed it in big stocks of enzymes that I've just mentioned, and then carried out all of the experiments in one go. So to be short, it's an incremental process.
Okay, and then similarly to decide where to put the transcription factor binding site, or
So 2 things happen in parallel because we're quite eager to get the sensing and the directed evolution going. We arrived at something which worked and then didn't touch it. I think I talk about it in chapter 3, and this might take me some time to find. But we tried a couple of orientations and we found that you need the nicking site to properly overlap with the operator site rather than just being next to each other. And as long as we worked, we just proceeded and had to live with the system. And then in chapter 3, because in parallel I was experimenting and thinking how I could improve it. I outlined quite a number of ways this could be done. However, we did not dare disrupt the system, which somehow worked, in fear that we would have to re-optimize everything. And this is a big concern with these mixes, is that if you change the concentration of one of the enzymes, because it impacts everything in the system, most likely you would have to then re-optimize concentrations of all of the templates, and maybe vice versa. I'm now curious where I showed this, but maybe it will take too much time.
Okay. So if you were going to estimate then for, I guess maybe two questions in one, for a new student joining the same group and for another group that wants to take your technology and use it in their own lab, what kind of a setup time would you imagine before?
So if it's just replicating the same experiments, it would be minimal, as you can take the recipes which are ready, and then one would maybe have to go through an iteration of varying concentrations of each of these components a little, just to get the sense of how the effect propagates. If the improvements were to be applied, this very much depends on the transfer of knowledge. I found it sometimes that over time I had the understanding of the dynamics sinking in, and then discussing it with Guillaume, who said, yeah, I thought of this half a year ago, these are the templates I've already designed, and you're like, okay, I thought of the same thing. And you see that sometimes bouncing ideas or exchanging them really helps. If one were to do it just based on a paper, it would probably be reasonably challenging. I'm sorry, I cannot find straight away the design which didn't work.
Okay, that's okay. I think it was just a sort of general question about effort, right? I think a thesis manuscript always hides these things of the one experiment that took several months and the one experiment that was very quick. You don't get as much of a sense, so that's just what I was after there. I also wanted to know a little bit about the systems that you combine, where you add LAC-I and TRIP-R in one, or you invert them in different combinations. So beyond directed evolution, which I think you covered very nicely what use they would have in a selection system, can you think of other things that would be of interest?
So, I mean, what we have here is an analog adder, which in itself is quite cool. I mean, here it's just addition and subtraction. If we add exponential amplifications motifs, we can have something more exotic, maybe. And I mean, this is more of a theoretical application, but I do not see a reason why you cannot add more sensing templates taking in different inputs. You could have several switches running in parallel and then integrating into some combined computation. You can think of a situation where you can have an incoherent feed-forward loop. And in principle, I think this is the exciting thing about working with the DNA layer, is that you do not really see any limits to what can be done there. And therefore, I think if you combine this with some real-life inputs, suddenly you unlock all of this potential which has been previously reported. Because analog adders in DNA have been reported quite extensively, and in fact it's something crazy like computing complex algebraic functions, which I did not think was feasible, but it is reasonably straightforward if you look at the flow diagram straightforward for long enough, and I mean it takes a whole page. But we did not go there because it has been done before.
Okay, I think that's all from me. So I'm happy to pass back now. Thank you very much.
Thank you.
Thank you, Karen. So I will go on further, as I'm also a rapporteur. And first, I'd like to thank you for the invitation. I was really pleased. This is not my field at all, so I was quite pleased to learn about these ways to actually screen enzymes, because I've been doing a little bit of enzyme evolution before, but the screening was really completely different. So that's quite interesting for me. So just some parts about your manuscript. The manuscript was a bit dense for me at the beginning, but when you reread it several times, which I had to do, I think it comes through very nicely at the end. This probably reflects the fact that the way of implementing the things are actually quite complicated. While it seems, when you read everything, that it seems quite simple, but actually I believe it is quite complicated. And I believe you probably suffered a lot into setting up the system. So that's a good sign, because it tells you that you've been persuasive on implementing this thing. And when you were talking with Karen about the fact that it might take some time to implement it in another lab, this is quite interesting, because I believe, it does not come through very quickly to anybody who would not know how the system works. Anyway. And I appreciated also the small summaries at the end of each chapter, so that I could make sure that I did understand exactly what was happening inside the chapter. So the first achievement for me was really interesting. The fact that you could use the system at 37, which did not. I mean, this of course is evident that you need that if you want to do further screening of enzymes. You need to be in conditions where enzymes work, and temperature is of course one main problem. So in general, it's a bit related to the question Karen asked about the sensitivity of the systems, because I have, what was surprising to me is that there is no standard deviations on the point that you have in your figures. And this tells me, so, and there was some little sentence somewhere that you always take the highest switching point, the highest CQ. And I was wondering, why do you take the highest each time? And do you actually perform 3 times the same experiment and measure a standard deviation?
So throughout this work, I did not focus on making the system most efficient possible, because I preferred working with something more stable so that I could demonstrate the proof of principle. And the sensitivity, as I mentioned, could be improved at least by increasing the amount of the concentration of transcription factor many-fold. And in fact, we rarely did exact duplicates and triplicates, because these would be technical repeats, as there is no biological variations we would expect. The only time I've extensively done this is, I think, for this curve, and then I computed the standards. So here a triplicate was performed. I computed the double standard deviation for each point, took the highest one, and then applied the percentage this double standard deviation represented relative to the 1/CQ to all of the other plots I've presented here, just to have some idea of the uncertainty. But my main claim is that I believe, because I have so many curves which show some trend, we do detect something. And in terms of how I convince myself that this is stable, if I could stop the slideshow and open a different one, which would look a little bit undercooked and dense. I apologize for the technical
It's okay, don't worry.
difficulty. So as I mentioned, for example, when I tried making these final pretty plots, I usually did about 10 experiments, gradually progressing. And what I find quite satisfying is that after 3 years of combining these mixes, I could sometimes consistently replicate the same point within a minute. So this is the slightly raw data, which I guess I would be more shy of demonstrating. But for example, here, with a variation in conditions from experiment to experiment, which are plotted in a series of 3, you see same trends. And if I look for something maybe prettier, here the 2 samples pretty much overlap for some regions of the curve from one experiment to the other. I cannot tell you off the top of the head what I changed between them, but I think it also illustrates the point of this gradual evolution of the reaction mix. So no, I do not have a robust statistical proof for majority of the plots I've shown, rather than just propagating the error I've estimated once.
Mhm.
But I believe, because I demonstrate such a collection of experiments throughout the thesis, combined, they give us some confidence that all of these trends are not an artifact.
No, I'm not telling about the artifact, because I do believe that there is no artifacts in what you've written. I was just wondering, because you actually explained that for each of the experiments, you come up at the point where you have the most sensitivity between the fallout and the amplification. That's the point where, of course, you can the bistable switch is operating either in one condition or the other, and that's where it gives you the most sensitivity. And so, and I was wondering whether this actually could influence the fact that you end up with, in sometimes, having points that are for example, in one of the figure, can't remember which one, you have some points that are dropping at the end of the 1/CQ drops at the end of the curve.
Oh, sorry, you were talking about that, and I wasn't sure what you meant. But are you talking about this?
Yeah, for example.
So,
and you have, some kind of a trend of diminishing things at the end, and actually the blue is actually dropping to zero. And I was wondering whether this is due to the fact that your conditions are extremely thin in terms of concentration, in order to switch to one to the other, and this could influence sometimes your data.
I apologize for misunderstanding your question previously, but whenever we observe this usually happens at the extremes of the input concentrations, and we usually attribute this due to disruption of the system. So in this case, we're reasonably confident that large excess of the enzyme, and hence protein concentration, and the buffer it carries with it, just disrupts the switch.
Mm-hmm, mm-hmm.
So I think this is not to do with the sensitivity of the system and the way it is intended to work, but rather disruption. And throughout my thesis, you also see that on the edges, some saturation occurs, as for example, also in this plot, in this plot over here. Unfortunately, there is an animation underneath, so I guess I could slideshow again.
There was also one of the first curve. I think it was on slide 24.
Where on slide 24?
Maybe not this one. No, maybe before. Can't remember. Actually, the first curve where you demonstrate that it works at 37.
The Lakai related one.
Yeah, this one. I was surprised because for me the last concentration of Lakai, at 110 nanomolar. The difference between the point before the yellow one does not translate into a high difference into the 1/CQ. Whether if you look at the fluorescence curves, between, for example, the last green and the yellow and
Yes.
the red, this does not translate very much on the graph. It doesn't seem to translate into high differences.
I agree. This I found quite unsettling, and I double-checked it several times, and I could not find the reason for this, I would admit. I think the problem here is twofold. We have the 1/CQ inversion of this axis, and also we have logarithmic scale over here, which somehow disrupts what we see. In fact, within the thesis, sometimes when I try demonstrating the concept, I do not use 1/CQ, precisely because this drop does not seem very significant compared to this one. But I think it is due to the fact that at higher numbers, this effect starts being less significant.
Okay.
And the part about the logarithmic scale is irrelevant. I apologize. I just said it to throw everything at you.
No, but that's important because for me it tells that actually your endpoint might not be that stable either. That it is the endpoint of the curve. I mean, is there a threshold at one point where you decide that you actually reached, for example, you can see it at the end of the curve. It doesn't change very much on your 1/CQ, but does that mean that even big changes of the CQ, when the CQ is actually reaching, is above the threshold that is really high, that it doesn't count anymore? The infinite time is actually at a threshold you can consider it 0, period. After a certain threshold of time, I don't know, here it would be around 8, you consider that whatever the time is, it's actually 0.
This could as well be the case, and the more I'm looking at it, the more it could be that indeed the system saturates here and whatever happens later. And this is another possibility. I'm not sure if I demonstrate this in the manuscript, is that extremely high concentrations of the transcription factors, especially LAC-I, quench the entire system and seem to disrupt the switch, not due to the binding of the protein sensing template, but rather due to presumably indiscriminately binding everything, and because transcription factors are known to sort of bind and scan DNA for the operator site, because that's how they find it. And indeed, this is what could be happening here. I would admit, at first, this experiment was carried out to normalize a stock of LAC-I and check what concentration is usable in the experiments, and then later we realized that with this we could also demonstrate that as you keep on adding LAC-I, you repress the switch. So maybe if I were to repeat this, I could go higher and look at that as well.
Okay, and you mentioned that you have a KD for IPTG or allolactose, I can't remember, that is 5 times lower, and you find that quite reasonable. I was wondering whether you find it really reasonable to be 5 times lower, considering that on the others, can't remember which one, you really end up with the same value of dissociation constant, or was it just, so can you comment on the fact that 5 times is really imaginable?
This is where we start bumping into the multidimensional constraints, and the concentrations of the transcription factors which we used throughout the experiments were primarily determined by finding a concentration where some noticeable repression occurs, as I've seen, for example, for LAC-I. I hope you agree with me that you plateau off. And I've picked one of the lowest concentrations at which this happens, because at highest you start disrupting the switch. In theory, you can shift this KD left and right, as I show for tryptophan repressor, by increasing the concentration of transcription factor. And in fact, one of the works we were inspired by, which uses transcription factor to interfere with RCA, demonstrates extreme sensitivity, because there they use a ratio of transcription factor to the DNA template of something like several tens of thousands, or I think to 1. We could shift the KD for LAC-I in theory by increasing the concentration of LAC-I. However, for some reason, we bump into the constraint of LAC-I disrupting the switch, as you just pointed out, which is why we settled for what we had. In the way where I say I find it reasonable, I guess I'm trying to say that this is something you can work with. You still detect concentrations which you can dissolve in water, you can add, they're easy to handle, they are non-disruptive for the system.
Okay. Okay, this. Okay, then we can switch to the in vitro evolution, and this part is again really nice. For me, it was a nice idea and successful in implementing. I was actually expecting that you actually did some work on the tryptophan enzyme on in vitro evolution on it, really doing implementing. I understand that the thesis is always restricted in time, so you cannot do everything you'd like to. and probably this signs that the system has been quite long to implement, and I really acknowledge that. But I was, you always end up with being a bit frustrated when you set up a nice system really, and you cannot use it really to perform the in vitro evolution anyway. So that on the manuscript page 70, that's the figure 66.
If you just give me a
Sure,
second.
yeah.
So I think on this manuscript 70 is 90. Is it? Yes, figure 66.
Okay, I'm trying to put it myself. Page 90, you said. Yeah. Why did you pick the 2 millimolar? And I didn't understand really why didn't you pick exactly the what is happening at, it's you have a strange behavior for me, which is completely near, and then there is the cutoff at 1 millimolar, and then you choose 2. So could you explain the rationale in here?
It is rather arbitrary. And you'd find that for quite a lot of situations, although we observed some trend, there was no clear point which was marked as the cutoff, so we had to make the decisions ourselves. And 2 millimolar represented a concentration where the diminishing activity was not too bad. We expected the yield to not be absolute, and therefore we wanted some excess of indole compared to 1 millimolar of L-tryptophan, which is what we used as a positive control for our sensor throughout the rest of the work. You would see that pretty much every experiment to do with sensing has as positive control 1 millimolar of tryptophan. So I hoped that with 2 millimolar indole and an active enzyme, we would get there. And I seem to recall, I mentioned somewhere a calculation that 2 millimolar was reasonably convenient to add to a mix using a water-based indole stock, which we also found important. Because I think even at 2 millimolar, you need to use quite a large volume of the water-based stock, and you only have so much free volume in your mix because you need to add other components as well.
Okay, and do you have any idea what is it? Because you seem to imply that it's an indole itself that is inhibiting the reaction. Do you have any idea for that?
So here it was indeed the case, and it was specifically the case for the MIR-based buffer. If I recall well, I demonstrate some more robust behavior for the isothermal prime amplification buffer developed by Rémi Siskind. I did not dwell on it too much. My immediate thinking is that indole can somehow interact with DNA due to the double ring structure, which is reminiscent of guanine-adenine rings, and it could somehow disrupt the interaction between the templates, or it could disrupt the interaction between the enzymes which specifically interact with the base pairs, such as the nicases, and have some effect, or obstruct the polymerization. To be honest, I'm not sure.
Okay. So for me, so what is I would now maybe switch to maybe more general questions. So now, if you, the system you describe and really needs at one point a transcription factor, so that is that it can translate the molecular signal from the enzyme into something that binds DNA. How would you implement it if you don't have that? The vast majority of molecules do not bind to transcription factors.
I do. So for someone who comes from synthetic biology, which was bacterial based, I would claim that there is a database of if I could defend the transcription factors for a second, there is a whole database developed by Jean-Loup Fallon, which lists transcription factors which have either been verified or are speculated to be present. And if we think about evolving natural compounds, most likely, if it's used by the organism, they sense it somehow, either directly or indirectly through metabolic conversion, and we demonstrate that we can do in situ metabolic conversions as well. So for example, if we wanted to sense indole, we could also produce tryptophan and therefore claim that there was indole at a certain concentration. And then there are also reports of artificial transcription factors with works, for example, by Mark Echelon, which demonstrate that you can minimize it, invert the logic, and so on and so forth. Transcription factors sensing artificial molecules such as bromotryptophan or fluorotryptophan. Alternatively, of course, you can use aptamers, which at first seemed to us to be a less robust option due to a reputation of having higher KD. When I was writing out the manuscript, I noticed that, especially for tryptophan, indeed there is one with a KD similar to that I've observed in my system, which I remark. But then as soon as you start functionalizing aptamers by adding fluorophores or some functional domain which allows self-cleavage, you usually strongly disrupt the aptamer itself, and therefore I think despite all of the progress, it would have been a little bit more difficult to work with, because you have within the aptamer both the capacity to bind and then do something else, and you would need to balance the two. Whilst with the transcription factor, it just senses and it binds, it does nothing else. You control all the processing through working with the templates, as I show in chapter 3 with, for example, chemical modifications and alternative designs.
And do you think it would be easy to actually set up, because that's the answers were of Jean-Loup Follon, that was what I was expecting, of course, because that is something which is which he has used in vivo first, but in vitro after, and with success. So yeah, you can use metabolic reactions. And but do you think in your system, so how, now that you know how long it takes to actually set up the system from scratch, how, if you had one or two enzymes more that would convert one of the product into something that binds the transcription factor, could you estimate the time to set up this entire system for detecting?
It is difficult to predict without knowing what the enzymes are and what temperature they work
Imagine
with.
you have the enzymes ready. I mean that you would not have to purify them, and everything would be ready.
I understand. What I'm trying to say is that if these enzymes interfere with DNA, or if one of the intermediates somehow binds other proteins we're using in the system, then we're in trouble. Best case scenario, you just add them in, they work. You also need for them to be compatible with the reaction buffer. I mean, our reaction buffer is quite close to physiological pH, or pH within the cells, I would claim. Salt conditions are not too bad. This would depend. And then there is always the wild card we've discovered with the Pyrococcus furiosus, which gradually disappeared from the final plot I've presented, because unexpectedly we discovered that it strongly and irreversibly binds DNA once you heat it up. I would not be able to predict this without trying. I looked at the structure reasonably carefully. I'm not a structural biologist, so it was challenging. I would claim that it is stable due to strong ionic interactions, and once you unwrap it, it prefers binding to negatively charged DNA instead. So long story short, unpredictable. Best case scenario, you just add them, they work. This was the case with the E. coli, tryptophan synthase subunit B, which we got first. And we were quite... it took us a much longer time to carry out independent controls, whilst the preliminary in situ detection more or less just worked. I mean, give or take the indole problem, which was resolved within half a week.
And lastly, if you, that's about the same question. If you think of biosensing applications, which are, of course, and you talked about them in your manuscript, would you imagine detecting real samples where you end up with having urine or blood samples which are full of a lot of different things and so on? Would you imagine that it's feasible to detect something without further purifications or dialysis, or, you know?
So I'm blessed with a set of colleagues who are trying to do precisely that with microRNAs. And in general, from what I understand, you need to carry out an extraction process, as even low concentrations of urine and blood strongly disrupt the system, which is built to be sensitive. So short answer it would be challenging, and it would probably take another PhD to figure out a way to just immobilize the molecules of interest, or to somehow make the system robust enough to tolerate a 10% dilution with whatever you wouldn't want. An interesting thing, for example, is that when I was looking as to why the Pyrococcus furiosus tryptophan synthase could be disrupting PCR, a lot of articles came up on blood components, and in particular, I think hemoglobin being also toxic for PCI and maybe DNA amplification in general, as it strongly binds DNA. So blood should be especially challenging.
Okay. Okay, well, I think I'm done as well. Thank you again for your invitation. Thank you for the manuscript and your presentation, which I didn't tell you was very clear and very well done and very informative as well. So thank you very much. And I will leave the talk with Philippe now.
Yes, so yeah, I completely agree with the presentation was very clear and very helpful, and thank you for this. So yeah, when you look at this work, I find really fascinating that it works at all. You put so many components together, and we know that it's one thing to draw diagrams, but then you need all the physical, chemical conditions to be compatible, and you also need the parameters to work together. And I think the questions of my colleagues also reflect this fact. So I have a few questions on design, but before, the only little thing I miss in your introduction is the positioning of your strategy compared to other continuous evolution systems. There are other people who try to develop systems based on in vivo or in vitro strategies. What are the advantages or disadvantages compared to what you are trying to achieve? Like I think about the system of David Liu, for example.
So, off top of the short personal answer is that I was excited about the modularity of the PenDNA toolbox, so I mostly jumped on an opportunity to work with Unique and construct some plug-and-play fundamental tool, which happened to be relevant to the particular pipeline developed by others. And I really focused on the interface rather than contemplating on the entire cycle. We can think immediately that this is quite a strenuous batch process, as you need to do a triple emulsion first to produce the beads, then to express the protein, then to do PenDNA. And in addition to all of these overlaps of conditions within the PenDNA sensing, you need to amplify just the right amount of DNA so that you can express the protein, but it's not in excess so that it is toxic. As Ramiz reported, you need to produce just the right amount of protein so that, again, it is not toxic to the pen, but it is detectable. And then get the pen just right and be able to cleanly extract the library so that you can go for the entire cycle. So the process is quite finicky, but I believe it gives you a high level of precision, because on one hand you need to get these conditions just right, on the other hand you do have control over what you do. And for example, could you remind me of the David Liu system you're referring to so that I didn't get it wrong?
Oh, it's in bacteria. There is a set of plasmids where, well...
Is it the PACE-related one? No?
Yeah, I think so.
Okay. So there we can, for example, think of an expression bias because it is done in bacteria. Here you would not expect as much of an expression bias if you have sufficient quantity of IVTT, no matter how badly optimized your gene is post-mutagenesis. You would expect to saturate the bead to its full capacity, and therefore every time you screen you have equivalent concentrations of proteins within your screening chamber. You also have the benefits that if it's something toxic for the cells, it probably wouldn't work too well. So what Adele has found during her project is that nicases, as you would expect from them, nick bacterial DNA, which makes it quite challenging to grow the cells, and you need some accessory plasmids which protect it to some extent, but cells really try to mutagenize the expression cassette and get rid of it. The advantage of PACE is, of course, that it's continuous, hands-off, and you can have remarkable results within a day or two with little interference by the user. But there are also the constraints. And despite the fact that once you got it going, it's all nice, you might remember that the system involves constructing a phage, 3 vector plasmids, which are probably also quite demanding on the cells, and then getting the turnover of everything just right so that you do not flush all of the cells, yet you have some turnover. So there are pros and cons for both. Ours is technically probably more demanding, but it is much more versatile.
Yeah, I agree with the fact that it should not be too continuous, because here you can have intermediate quality controls and you can stop the thing and restart it. So I can see this advantage. So I have a more specific design question. So for example, on figure 42, but maybe we don't need to go there. So you have this strategy with a transcription factor, and it competes with nickase, if
yes.
I understand.
Yes.
So how did you design the sequence? I mean, how did you need to tune this competition? Did you pay attention to that, or did it work? I guess.
Not really. You do not have much choice, because you have an operator site and an nickase site and an output, and you need to combine them somehow, and there are only so many meaningful permutations. So I think somewhere in the manuscript, and unfortunately I was struggling to locate it off the top of my head, alternative to this, the only, you could try moving the recognition site to our right and have the nicking site here, but I would not see the point, as you would have less overlap. You could also move it by 1 or 2 nucleotides. It never occurred to me because I wanted maximal interference. We've tried locating it on the other side, in which case the nickase recognition site is just neighboring the operator domain, which was not sufficient. And I think with the NT-BST and BI, this is about it. What I think would be quite cool, and we've tried this and showed that you can do this, is locate the nickase recognition site here, over here, so that it cleaves itself off, and then you have every time the same tail of the functional domain and the 4 or 5 base pairs of the nickase recognition domain. And also because it cuts just adjacent to the site, there is no context dependence, whilst, as I've mentioned, we've discovered that these 4 base pairs, which we originally thought did not matter as to allow different operator sites to be put in fact strongly affect the nicking activity.
Mm-hmm. Okay. And yes, so when you combine signals, like in figure 53, it's a very stupid question, but how is integration done? Is it multiple transcription factor on the same site, or is the integration done at the level of the pen DNA toolbox? So
maybe it wasn't too clear, but the way, maybe I've missed it in the text, but no, I do specify that we combine PST LAC-I, which is this guy, with, I pointed the wrong one, PST LAC-I, which is this guy, with PST tryptophan repressor, which is this guy. And we played around with concentrations to get them just right, because they have slightly different efficiencies. So it is 2 templates, which then both, as you can notice, have exactly the same output, just a different tail, which is determined by the operator site. So both of them have exactly the same -2+2 output, which can then go and bind the pseudo template or the autocatalytic template. Therefore, the integration is done on the pen level.
Oh, okay, yeah. Yeah, because for this module, you don't have the drawing where you actually draw the pen toolboxes. For me, it was hard to...
I thought...
But it was written.
I hoped that I summarize what the switch is somewhere at the beginning, where I introduced the basic modules.
Yes.
And then I kind of say, okay, and as before, plus the switch. So here, I specify that it's always these three guys, and then everything else feeds into them. So when we go to our multiple input implementation, it's this assembly of these three guys plus the enzymes, and then the two PST, which act together with through L-tryptophan and IPTG.
Okay. So now I have a few questions about the dynamics of your system, which echo some other the former questions. So for example, on figure 44, I'm generally wondering about the steepness, the cooperativity of the response. So for example, here, that was discussed before already, but this slope, what is it? Is it 1? Is there some, the Hill coefficient, is it typically higher than 1? Is there some cooperativity? And is that something you can control? Because I can see, for example, on figure 50, it seems that the slope changes. Do you understand those things?
This is something which did not pay to my attention towards, and I think for this first figure, we did not have the Hill coefficient. Whilst we did extract it, once we saw that indeed there is a sharp shift. I do not have a comprehensive set of data to explain all of the possible behaviors. However, in this case, what we see here, I think, is hypersensitivity. So below a certain threshold, all of the source is consumed and completely quenched by the anti-trigger. However, this amount of source is gradually building up between the samples, and at some point, once it exceeds this threshold, it just goes and it activates the switch. Whilst in case of the non-killer system, the process is somewhat more gradual, because you're gradually feeding into this autocatalytic template, and there is this in-between state where some of the trigger is still consumed by the pseudo template and maybe the reporter template, something I talk about, I think, at the end of chapter 2 with respect to the reporter template, while some of the trigger is starting to participate in the amplification cycle, making it less steep. So there is that. We can control the KD. The KD is somewhat linked to the KD of the transcription factor, and you can just move a little bit up and down the sigmoid by varying the concentration. It is indeed conceptually quite challenging to represent these things, because you have the transcription factor allosteric response, which is following a sigmoid, which then is projected onto the response of the system to the binding of the transcription factor, which is also sigmoid, which then translates into the activation of the system, which is also a somewhat, well, in ideal case it's bistable, but in practice it's another sigmoid, with the response of the curve itself, which we observe also being a sigmoid. And you can see how we're going down the inception film
what happens,
plot.
I mean, that was shown early for people trying to do synthetic biology of regulatory system, but if you have one everywhere, it will stay one, but you multiply. So when you have many sigmoids, but they typically have a high cooperativity, at the end of the chain, you have a very steep response. But it seems
so in terms of cooperativity, I can't exactly tell why it behaves a certain way. For the hypersensitivity case, we speculate it's that behavior. For the less steep guys, we think there is more of taxation on activation of the switch, which linearizes it. And in fact, although I think the exact data is hidden, but for example, for the isothermal primer amplification, the switching curves are much flatter, let's say. And I mean, they're still reasonably steep, but they're not as steep, which suggests that because the isothermal primer amplification buffer is less efficient for the switching and for PEN, you therefore sort of decrease this cooperativity response.
So I have two related questions still. For example, on slide 20, we see the rise of the autocatalytic response. So what determines the rate? Yeah. So what determines the rate of the growth rate of the autocatalytic system? What is the limiting step in the?
The limiting step is defined by the experimenter. It can be different things because this activity requires NT-BST and BI. It requires interaction between different DNA strands. It requires activity of DNA polymerase. And then to some extent the PT has an effect because, as I mentioned, even when you have the autocatalytic loop, it is still able to consume the trigger. In early stages, some of the trigger can also go to the reporter template, to primary producing templates if they are present, and so on and so forth.
Well, yeah, I agree that everything matters, but is there a bottleneck? Or do we know? I mean, maybe we know.
I do not. The bottleneck is the so when I optimize these guys one by one, and I would expect the first guy I optimize to become the bottleneck by the time I've optimized everyone else, because once you optimize the next step, the previous optimizations become less relevant. This is up top of my head. If you give me a second to meditate on it, I might give you a better response.
Maybe later.
But when I was thinking of what could I improve and how could I make it sharper, something I did not apply to the systems I'm reporting, although it is shown in chapter 3, is really the taxation of the downstream modules. So in my case, the affinity of the trigger sequence is the same for the autocatalytic template, for the primer producing template, and I think the reporter template affinity is even higher. Therefore, in these early stages of exponential amplification, where it's critical to get going and get up this curve, because you're still at risk of being quenched by the PT, the trigger is also sucked off to open up the reporter template and to start binding the primer producing template. So if those were to shorten and have less complementarity to the trigger, I think I could have sharper amplification.
Okay. So now about the performance. So what is the lowest concentration of a small molecule that you could expect to see?
In my case, I think this is reasonably self-evident from the plot. But as I mentioned, I never attempted to optimize for sensitivity because I wanted a robust system with which I could play around, reorient, plug and play the modules. So here, with respect to L-tryptophan, we see that we can probably distinguish concentrations of, well, even 10 micromolar and, well, actually, sorry, this is negative control, so maybe we can't. Around 10 micromolar, and then if we go at detecting L-tryptophan and then translating it into PCI, here we go to about 62 micromolar, which I think is reasonable concentrations for directed evolution, because this is the quantity you can produce with 1 micromolar of the protein, which you can produce in TXTL and then immobilize on the bead. But in principle, as I've mentioned, there are works which specifically looked at optimizing sensitivity. And there are two approaches which are quite obvious. One on the pen level, which is making the system as sensitive as possible by approaching this bistable point, making it less convenient to work with, and maybe also slowing it down. And on the protein sensing level, it is vastly increasing the concentration of the transcription factor relative to the protein sensing template, because then, although you're eventually constrained by the response curve of the transcription factor to the ligand, you can, of course, go down this curve and push for the natural limit of the transcription factor.
Okay. So now I have a few last perspective questions. I have a very simple question. So on your last slide, you run the thing in open beads, if I understand. So does it stay inside? Does everything stay inside, or there is nothing that leaks out?
So to be clear, I mean, I didn't. The slide is a little bit messy because it is a teaser. In this experiment, I incubated beads in bulk in an IVTT mixture, containing DNA, which was separate from the beads, so that ECB coupled with snap tag, which allows the immobilization of the bead, would be expressed. I then took out the beads, washed them extensively, and added different quantities to to a total volume of 10 microliters of my pen DNA reaction, and same for GFP snap. From this, I would, from the fact that these guys work, as well as some of the controls I'm not showing, I would infer that I do not transfer anything else from TXTL, which could be problematic. And at the same time, because I detect switching when there is ECV snap, which is quite similar to when you just add the protein in bulk without the beads, I would claim that I do transfer the protein. I'm not sure if I answered your question and whether you were maybe asking about emulsion, which is not there.
Yes, well, maybe I don't understand enough how it would work in the end.
I did not attempt any emulsion work, and I'm quite satisfied to just claim that if my target is immobilized on the beads, my sensor can still detect it.
Okay.
Because I would claim that Remy Siskind has proven that you can amplify the DNA on the bead, solidify the bead, wash it, express the protein, and immobilize it, and do all of these steps in emulsion, so I did not replicate them. The only concern I had is that when you transfer to emulsion, your reactants or your products might leak. And we found that it is the case for indole with a partitioning coefficient of about 1:1 oil to water. But we think this would not hinder selection because, in fact, the droplets would compete for indole, which would be sucked out of the common media, whilst the product L-tryptophan would not escape. This is something I've also checked with this colorful micrograph. And I think my PDF viewer is frozen, but there is a micrograph in part 2 showing just that you can have droplets with L-tryptophan, droplets without, and only droplets with L-tryptophan switch on when you incubate them together.
Mm-hmm.
I'm not sure what happened to my PDF viewer. I'm sorry about that.
Okay, I will jump to my last question in interest of time. So it's a philosophical question. So we see biology as something fine-tuned and robust, and we think it's because there has been a lot of evolution. And you have tried to put a lot of processes together artificially. So are you more surprised or less surprised about the fact that you are alive?
I think I am less surprised at this point because I had the opportunity to go into the detail and see how it works and see the approaches by which you reach these little islands of stability where the magic happens. Coming back to my undergraduate experience, in fact, my first semester or my first couple of semesters I found quite depressing because it seemed that life is in this constant state of decay and chance. And in fact, this is the reason I gradually migrated more towards in vitro simple DNA reaction rather than going onto biomedical sides. And in the spirit of the Gulliver unit I'm currently in, I find it hugely rewarding that the PCR reactions, which I did in my first week in the lab during my bachelor's on the count of hundreds, because my project consisted of just PCRing a bunch of things, I now consider non-trivial at all, and I think it is an extremely complicated process which is never guaranteed to work.
Okay, thank you very much.
Thank you very much, Philippe, and I will leave the talk now to Yannick.
Thank you, Gilles. And first of all, I want to thank you all members of the committee for participating in the defense and also doing the effort of going deep into the understanding of the work of Vasily. And I know by personal experience that it is not always easy to understand what's going on in this system, this complex system that we try to build, and they are not very intuitive. So thanks a lot. Going on a little bit on the philosophical side, after Philippe, yeah, indeed, as was very clear from the presentation of Vasily, and from the question session, the big challenge here was to assemble complex molecular process in vitro. And it can be seen a bit weird that indeed we see all those living systems which are molecular. They are even much more complex than whatever we have attempted here, and they are all over the place, and they work very nicely with genetic regulation networks with thousands of nodes and metabolic networks with hundreds of different molecules. Everything going very smooth. And on the human technology, we also have very complex systems built out of small elements in electronics, and we are very good at assembling complex mechanical systems from parts. But apparently, we are not that good at doing the same for molecular in vitro systems. And so this is, I guess, something we are currently trying to learn how to do, and how to make this work, how to understand them, and to develop some intuition, or also some tools to go on in this way. And it's especially challenging when you include the because those systems are nonlinear, as was also mentioned during the question session. You have exponential loops, you have bistability coming in, and so as soon as it becomes a chain of nonlinear events, it's extremely difficult to manage them by intuition, and even also to manage them by mathematical predictions. So that's probably the system that was demonstrated today by Vasily is one of the most complex in vitro molecular multi-step process, especially if we consider that what the challenge here was to bridge different chemistries, right? Going from small molecule chemistry to DNA biochemistry and to replication process, and that's relatively. I think it's relatively unique. If you look out in the literature, there's not that many examples of multi-stages process that can do that. So so that was the scientific and philosophical remark. Besides that, I want also to congratulate Vasily for the quality of the presentation, and I think I'm in the position of giving this congratulation. I mean, I could not do the congratulation for the quality of the work because, of course, I was much involved in the scientific work. But for in the case of the presentation, it was a much more personal preparation from Vasily. I was lightly involved, but relatively lightly, and so congratulations for the very nice the presentation that I thought was quite interesting and well made. I also very briefly want to transmit my acknowledgement of all the efforts besides scientific research from Vasily for lab life, and in particular social activities and links, social links between the different members within the team, and but also within the more general UMR, the lab of multi-team lab as we have in France. And Vasily was always very much involved in creating ties and links between different people, and I think that was very nice. Just to conclude, Gilles mentioned a bit of possibility of frustration, and indeed we are quite frustrated that we could not demonstrate the full loop, and we were probably quite close. Of course, there was COVID impact, like for everybody, I mean, but it took a couple of months out of experimental work for Vasily. Maybe without that we could have done it, but we are very close, and we will be looking for ways to go on this work and demonstrate the full completely in vitro selection or evolution campaign for a metabolic enzyme like this one, hopefully in the future. So with this, thank you very much, and we can, I guess, I will, Gilles, you will maybe say just a concluding word.
Yeah, I think we're done now for the question session, and I think we will be on a separate room. I don't know how exactly, but the jury will retire to deliberate.
So in fact, what will happen is that Vasily will move away of this room, and he will keep the same Zoom session.
So just to be clear, I will leave and immediately click to rejoin. This is not because I forgot to say something, but so that at your convenience you can let me back in.
Okay. Possibly we will, yeah, okay, let's do like that, that's fine.
Okay.
Perfect.
Hopefully see you at some point. And now we wait. Thank you for the 40 people who are still with me. It is impressive that you're with me through the questions. And I would admit from time to time I did look at the counter, and although I could obviously not wave my hands and be excited about it, it is rather rewarding to know that people are watching me and that I'm not alone. I will hopefully not wait too long until I am let back in. And maybe contact my family in the meantime to check if they managed to watch it all. And once we hear the decision, the stream will probably end, and I will start reaching out to people and then go back to the lab to celebrate. Interestingly, I had to do this from home because it is quieter and there is more space. But of course, I would like to congratulate Yannick in person, as well as say thank you to all of the people who have supported me at the workplace. The suspense. Exciting times. I'm discussing with a colleague who was the first one to find the David Liu figure illustrating directed evolution cycle, as we're now trying to find who used it first in one of the presentations, me or the colleague, because apparently this is a cool figure. Hello.
Hello, back again. First of all, the jury, after having deliberated in a separate room, has now decided to award you the title of Docteur de l'Université PSL, which is le Paris Sciences Lettres. If in English, of course, it's Doctor of Physiological Science, with no mention. You know that we in France we used to have some what we call mention at the end. We don't have that anymore. But all the jury wanted to congratulate you for the very nice quality of the work, for the very nice quality of your oral presentation, and overall for the discussion that we had a lot of pleasure with talking with you about this science. We all of us congratulate you for the very nice work that you produced.
Thank you.
I think we can applaud even if we are far away.
That's the minimum, but the best we can do.
Yeah, and again, I feel sorry not being able to be there. I know we don't have the choice, but it's a pity that we cannot go further into talking with you and having a nice chat after the PhD defense, which is the usual case. I hope you will be able to meet with your family and so on after that, even if the conditions are not already met for having a party. I hope you will have a nice one with the lab and the people around you.
Thank you. The plan is indeed to go to the lab later on and congratulate Unique in person.
Okay. Well, so I think we're done to get. I mean, the paperwork will be produced by me and will be sent directly to Yannick, and it should be done by the end of. The time I receive the different paperwork from the jury members.
Okay.
I think you send directly to École Doctorale, Gilles, but I will.
Okay.
Fine.
Okay. Well, again, Vasily, thank you very much for inviting me and having us on this special occasion, which was very interesting. And thank you very much for the long and lengthy but very interesting discussion about this in vitro system. And I will leave the jury members if they want to say a few words as well.
Congratulations also, that's very impressive to have put that all together. And I can imagine you are frustrated because of the COVID, because you could have done the full loop, but hopefully that will be done. People will remember you.
Yes, and congratulations from me also. I guess I've known you for a very long time, and it's been a pleasure to see how this has turned out and how great you've become. I think you're a really great scientist, and I I hope that you will stick with science, even for profit.
That is the plan. To stay working with emulsions, but for profit. I'm moving to Bordeaux, hopefully in a week, to start selling surfactant to stabilize emulsions.
You go to Emulseo?
Yes.
Okay. Great. Hopefully we will buy your stuff.
We'll have new products in the pipeline.
Okay, good. Okay. Well, again, thank you very much, and I hope. I mean, Bordeaux is not far from Toulouse. I have regular contact with Jean-Christophe Barret, so and we have microfluidic systems on site in Toulouse as well. So
it would
maybe
be...
maybe you have to come by and try to sell your emulsion systems. We do have a lot of people doing microfluidics here. So.
It would be a pleasure to meet in person.
I hope so. I hope so. Well, okay. Thank you very much, and again, sorry for this abrupt ending of the defense, and I hope we have other ways to reconvene.
It is okay. I would maybe also use the opportunity to thank the members of the jury to be so accommodating to do this over Zoom, and also fix the date at rather short notice due to some administrative constraints, and taking the time to read the opus, which is indeed quite tricky at first. I would also like to thank the online audience, which at some point peaked over 50. And there are still 30 people who persisted with us through the questions, which I find remarkable. And obviously I would like to thank my lab and Yannick for making all of this possible and managing everything until the end and through the defense. Thank you.
Thank you.
Thank you. Bye-bye.
Bye. See you soon.
Oh, I'm still streaming. I kind of forgot that one. So I guess this brings us to conclusion. Thank you for the brave 30 to 29 volunteers here with me, family, friends, also everyone joining at short notice, because I only figured out how to organize all of this last minute. Insignificant it may seem, but it did make a huge difference to see that there are still people here, and to know that all of you are watching, are interested. The responses of, yeah, sure, I'll stop in the middle of my day and tune in. Thank you.